Please welcome Professor Toner.
Thank you.
The floor is yours. You're welcome.
Thank you very much. Well, that about says everything about me, so I'll jump right in. Thank you for having me here. I've had a delightful visit so far. What a great facility, what great science is going on. So thank you so much for having me. My talk today is a bit of a hybrid. I've been thinking a lot about the future of synchrotron science for folks like me, scientists in the earth and environmental science subject areas. And so I'm going to basically do two things, or attempt to do two things. I'm going to give you some sense of the kinds of work that folks like me do at Synchrotrons. I'm going to try to boil it down into some core components, instruments we need, capabilities that we're interested in having in the future, and then I'll probably spend about half the time talking about one PhD student that graduated from my group recently. I think her dissertation really embodies a lot of the different tools and approaches that we're currently using now and places that we would like to grow in the future. Earth and environmental science is a collection of disciplines and subdisciplines. My count is over 100 of them. And they're going to bridge everything from oceanography to soil science, modeling, field laboratory studies, basically everything from earthquakes to climate change. So earth and environmental science is a useful grouping. The geology department at my university has gone to earth and environmental science as the name of their department and their graduate programs. But just, I want you to keep in mind that we are, the members of the earth and environmental science community, is a large and diverse group. Earth science itself, of course, is covering all of the spheres of the planet that we interact with: geo, hydro, bio, and atmosphere. We're interested in thinking about the properties of the Earth and its systems, how it evolves over time, and feedbacks amongst those systems. If you want to think about Earth and environmental science, then you need to add the human component in. The way that human beings affect environmental systems, the way we extract resources, create waste products. We interact as a separate and integrated sphere to a certain degree. So earth and environmental scientists do a lot of different kinds of work. Not all of us are synchrotron users, so what I want to do now is distill down that large and diverse group of researchers into those that are going to be using synchrotrons to understand their Earth materials. Earth materials are good uses, I think, of synchrotron capabilities because they are complex and they are heterogeneous, and we need really good tools for thinking about the characteristics of these materials. So this is where you have to know that this is my synthesis, my opinion of how earth and environmental scientists use synchrotron facilities. I tried to boil it down to what I think are a manageable number of factors or characteristics. And I think that these characteristics that we have as a community, as a user community, I think to a large degree agree with where synchrotron science capabilities are going in the future. Let's see if you agree. So the first characteristic we have as users is that we always have to be thinking about different elements. One element, even though iron is my favorite, manganese sometimes, it can't tell the whole story. And even if my goal is to understand iron chemistry, which it is often, I have to be thinking about the light elements, carbon, oxygen, nitrogen, the tender X-ray elements, aluminum, silicon, sulfur. These are elements that form basically our Earth's crust. These are the rocks and minerals, but they also get involved in biological systems and the speciation of metals. And then, of course, the hard X-rays are good for a lot of our favorite elements composing Earth materials or contaminating Earth's environments. The second factor that comes up a lot in my work is that we have to be bridging scales. And I think this is true in a lot of physical and biological sciences at this point. But we need to be able to think about nano, micro, and millimeter scales in the samples that we bring to the synchrotron so that we can understand larger scale processes. So often for us, that looks something like microbes to the planet and everything in between. And how this plays out at synchrotrons, of course, is that we, as Olga showed me today, we have a series of instruments that bring different spatial capabilities, different windows into our samples, and we can nest them so we understand the context of our materials and then be able to zoom in on the factors that are most important. And we measure multiple properties. So there are many instruments that measure one thing, and there are synchrotron instruments that measure many things. The X-ray microprobes are examples where we might do imaging, scattering, and spectroscopy all on one beamline. I really like those kinds of instruments. They let me get at the complexity of my sample. But there's magnetism and many other phenomena or properties that we might measure at synchrotrons. But I would say my group really falls into these three. And then we have this really large concentration gradient. My best estimate right now is it's about 9 orders of magnitude in concentration that would be very easily relevant on any given day at the beamline when my group visits. So for example, we might bring samples from the deep ocean. So we're dealing with nanomolar, 10 to the minus 9 moles per, say, kilogram of seawater that's been filtered through a filter substrate. Or we might be dealing with banded iron formations. These are rocks that might be 40% iron by weight. And we might bring both of these samples to beam time on the same run. And so this is a challenge that we face and that we deal with in a variety of ways, but it's something to think about. My group is not the only group that has this challenge. We are also interested in moving our science from where we are today to higher throughput observations. This is important for us to understand a complex and heterogeneous material. What is a representative sample? How do we have statistically significant and robust studies? So I would say at this moment, we would like to move in that direction more with the studies that we conduct at synchrotrons. It is a challenge. But I would say in general, over my career so far, we've gone from studying 1 or 2 samples in great detail with an X-ray microscope now to maybe we can do 30 or 40 or 50 samples in a study. That is progress, and we're greedy, and we want more. So this is a challenge that we've been thinking about a lot. And then data curation and analysis, I think, is always a challenge for my students. Most of us are not computer scientists. Most of us don't have skills in informatics, supercomputing. So I think as we move toward our goal of higher throughput studies and experiments, statistically relevant observations, this is something that we face as a challenge, I think, as a community. So those are my 6 factors. That's the Earth and environmental science boiled down for the synchrotron users. And here's how it goes. I would say we already have been doing good science, investigating multiple elements at multiple spatial scales and multiple properties. We're doing that now, and I feel like we're going to continue to need access to those types of resources in the future. And then where we're probably struggling is, there's more, but I wanted to make it a manageable number of factors. Concentration, especially on the low end, is a challenge. We would like to have higher throughput in our sample analysis. And I think probably if we get wish number 5, then wish number 6 becomes even more pressing. How do we turn that great volume of data into great publications and PhD theses and knowledge that people can use? So what I want to do, that was a very general synopsis. And now what I want to do is make it a little bit more personal and a little more specific. What I'm going to do is I'm going to tell you about one dissertation, Colleen Hoffman's dissertation. She's now a postdoc, jointly appointed between NOAA and the University of Washington at Seattle. She's doing great. Won an SF grant, 3 years. where she used 3 synchrotrons, all in North America, 5 different beamlines to study samples that were collected over 60 days at sea and that represent 300 samples and a 7,000 kilometer transect across a very important part of the ocean that I'll tell you about in a moment. And I would say this is an example that is somewhat typical of a day in my office at this stage. We're trying to really honor these larger studies that are available to Earth and environmental sciences while still bringing the really high quality observations that synchrotrons can provide. That is our niche. And the big science question that's on the table today, and often what I've been writing grants about over the past 5 to 10 years, is what is the iron budget for the global ocean? And just to give you some sense of where in Earth and environmental science we are located, we are thinking about the hydrosphere in the broad discipline of oceanography. My work often falls into something we call ocean biogeochemistry, and what that means is I study metal chemistry because it's important for organisms that live in the ocean. Not a biologist myself, but I always end up working in these groups where biogeochemistry is the general approach that we use. Iron's important in the ocean. Some of you maybe already know that. Like human beings, most organisms on the planet need iron as a micronutrient, and it turns out that because of a wide variety of factors, it's pretty limiting in about 1/3 of the global ocean. And where we care about this, where we think about it as being a limiting nutrient, is in the photic zone, so that depth of seawater where sunlight can penetrate, and where we have organisms, phytoplankton, that are basically the green grass of the sea, right? They're taking up CO2 to build their bodies, which makes them really important when we think about climate change, long-term climate and carbon budget for the planet. Their activities are limited by the availability of iron. So that's one justification for having this, pursuing this research question. It turns out that we don't know very well right now where all of the iron comes from in our ocean, where it gets to, whether it's bioavailable. So there are a lot of open, big questions. But one of the things that I want to talk about today is the controversial hydrothermal iron source. And this is iron that's delivered to the ocean through hydrothermal venting at the seafloor. It happens along a 60,000 kilometer volcanic mountain range, crossing all major ocean basins on the planet. And where you see these red dots, triangles, and squares are known and confirmed locations of hydrothermal venting. So this is truly a globally distributed source of iron to the deep ocean. Doesn't seem that controversial when I present it that way, does it? Is there a lot of iron being delivered at the seafloor? Yes. All the best estimates that we have indicate that hydrothermal venting brings as much iron to the global oceans as riverine input from the continents. And the way you can think about that here is hydrothermal flux in moles per year versus a river flux in moles per year. For elements that have about equal contribution to the ocean from those two sources, you get the 1-to-1 line. Here, iron is a bit on the high side of that. So if you can just imagine in your mind's eye for a moment the amount of iron, so in suspended particulates probably, every river on every continent delivering material to the oceans every day, that's how much iron you need to be thinking about being emitted from the seafloor through hydrothermal venting. It's a lot of iron. But the problem is, and the prevailing paradigm that I think is arguably starting to change, is that the iron that's vented at the seafloor can't escape the local area of the hydrothermal vent. So, how does this work? This is highly schematic, but it gives you the main idea here. You've got seawater infiltrating the ocean crust, interacting with rocks and heat. That water is chemically altered. It becomes something what we would call vent fluids. It's re-emitted at locations of a lot of fracturing as hydrothermal vent fluids, no oxygen, lots of dissolved metals, including iron in most cases. And when it hits the deep seawater, cold, oxygenated, you have great disequilibrium and particles start precipitating out. And so what this is showing is in a lot of locations, iron sulfide minerals are the first to go out of solution. And then through oxidative processes, redox reactions, iron oxyhydroxides of various forms then also precipitate. But the idea is particles form, particles settle. There's a lot of iron coming in through hydrothermal venting to the ocean, but it doesn't matter because it doesn't go anywhere. It can't get to where the phytoplankton need it. So that's the general idea. And the paradigm, certainly when I was a postdoc, was that these were abiotic reactions, they were inorganic reactions, and you could essentially assume safely complete removal of iron. So it wasn't going anywhere. But like a lot of things, when new instrumentation, new technology becomes available, you start to discover that paradigms don't hold up necessarily. And so what I want to show you is some of the first data that we collected, probably back in 2005 or 6 at the Advanced Light Source, that gave us some indication that the way we were conceptualizing hydrothermal venting really wasn't accurate. And so let me explain to you what I have here. These are scanning transmission X-ray microscopy products. On the left here, I'll always show them this way too. It'll be a transmission image. Here, this is a carbon elemental map. It's composed of a below and above edge image, and then the iron map. And what we found was that in hydrothermal particles, iron was co-located with carbon. And that's just not what was expected. And this was primarily an imaging technique, but of course, STXM allows us to understand the chemical speciation of the elements of interest as well. And when we went ahead and did that, we found some pretty interesting things. So these are element maps. This is showing the iron 3 distribution. I apologize, this is 2 microns. So these are sort of what I would call a big aggregate collected from the marine water column. This is the distribution of iron 2, and here is your carbon map. And what we learned through spectroscopy was that the iron 2 was co-located, actually all of the iron was co-located with organic carbon. And this went against our conceptual model for what these materials should have looked like. And those organic materials were materials that looked like they had been very recently living biomass, proteins, lipids, rich, gooey organic matter. And one of the things that we hypothesized was in an oxic deep water setting, in samples that weren't necessarily preserved to protect redox status of iron, we were still finding iron 2 correlated spatially with organic carbon. And one of the hypotheses that we put forward was that organic matter had to have thiol groups. It had to be having organic sulfur moieties present to really stabilize an iron 2 signal, preventing it from precipitating as an iron sulfide mineral or oxidizing to form an iron oxide mineral. Something about that organic matter was really important in the speciation of the iron. And this is just an example of some of the handoffs that we would then take samples from a soft X-ray microscope beamline and bring it to a hard X-ray microprobe line and try to look at different elements to understand the materials. So what you're looking at here is a large STXM image showing the very fractal nature of this organic matter. I'm showing carbon in white. 30 micron view of a material, of a particle. And then here, of course, at a larger spatial scale, this X-ray fluorescence map is 400 microns. And what I'm showing is sulfur in blue, iron in red. These are not the same aggregates, right? These were 2 different samples run through 2 different beamlines. But you're going to hopefully trust me that I chose to show you representative information. So yeah, the organic matter was loaded with sulfur. And we were able to at least have one piece of evidence to support our hypothesis that in the particular organic matter, organic sulfur was really important for the iron speciation. The other thing that we found in looking at these materials is that they were really filled with a wide variety of crystalline inorganic phases. And again, this was hard X-ray microprobe, but now we're doing diffraction. And I thought about showing you the table, right? The terrible, ugly table from my paper. But I decided to just give you a list instead. And what I wanted to show you was just the variety of different phases identified. There were probably many others that we didn't necessarily identify or had intermediate crystallinity, but zinc, manganese, iron, and copper sulfides were all identified, as well as other oxides of metals and elemental sulfur. So hopefully you're getting some sense of the complex grab bag of materials that are present in a given marine particle coming from a hydrothermal system. And I think what I would like you to appreciate is that the particles were not how we conceptualize them. They were not discrete, spherical, inorganic mineral precipitates, right? This is just not what we find in natural materials, and that is a discovery given to us by synchrotron observations. So they are always composites when we find them, organic plus inorganic components, wide range of crystallinity of phases, and surprisingly complex speciation of elements of interest. So I would say those discoveries led us to really question how we were conceptualizing the ability of iron to escape from hydrothermal systems. If we didn't know this, what else didn't we know? And so we then wrote a paper, this was quite a few years ago now, proposing something we're calling the leaky vent hypothesis. And the idea here is, because of the nature of the organic material, because of the nature of those organic iron interactions, yeah, we could actually propose a whole host of possible mechanisms to keep iron from precipitating into an inorganic phase and settling to the seafloor. And so then the question is, can iron get away? What chemical form is it in? If it does, can it be brought up to phytoplankton where it'd be relevant for the global iron budget? So, to summarize, we got interested in the transport potential of those particles. And we wanted to understand the solid phase speciation, and I would say right now, we're here. But let me connect the dots and get you to that point. So the thing you need to know about oceanographers is that we/they think about particles a little bit differently than maybe you do. And it's all based on operationally defined size classes. And so just to give you some sense of what this looks like, I mean, this was a paper in Science in 2017. I mean, we're still talking about, like, how big is something? But in any case, for the vocabulary that I'm going to be using in this talk, what I mean by a particle is anything larger than 200 nanometers. For those of you who are biologists or work with microbiologists, you know that often we filter to sterilize at 0.2 micron or 200 nanometers. And so anything captured on a filter that's greater than that size would be considered a particle. So that's a huge range of sizes, but that's how we do it. And then there's this middle ground where the oceanographers call it colloidal. Freshwater chemists have a different definition of colloidal, apologize for that, but it means you're something between 200 and about 2 to 20 nanometers. Why is it a range? Because it depends on how you do your filtration process and how you capture the particles. And then anything less than about 2 nanometers then will be categorized as soluble. So what you're going to see is I'll talk about particles and particulate, and I will honor that size class or that operationally defined definition there. And then anything in this box is going to be dissolved. So your nanoparticles are dissolved. But just keep in mind that this is the framework that we're dealing with. So the opportunity to look at transport potential of these particles came from a program funded by the National Science Foundation, but it is an international effort called geotraces. And we're going to look at a transect. Here's my 7,000 kilometer transect that I'm going to talk about. So one of the things that you should probably know is that transect crosses a really important part of the deep ocean. It is one of the biggest known hydrothermal plumes on the planet. And what you're looking at here is a helium contour map at 2,500 meters water depth. Okay, so a slice through the ocean at that depth. Where you're seeing the red and orange contours is where helium is. We use it as a conservative tracer for hydrothermal vents mixing in. So what you have then is a very large hydrothermal plume. The ridge axis is here. The prevailing water movement is this direction, but it is complex in terms of where currents go and how water moves. But this whole middle part of the Pacific Ocean is affected by hydrothermal venting. And just a close-up then to give you some sense, all of these yellow dots are stations where we stopped and collected data, samples. And again, the ridge axis is station 18. But I'll always try, I won't make you memorize the station numbers. I'll try to always refer it back to the feature of interest. So we can just jump right in. There's a bunch of important discoveries that came out of this field work that have nothing to do with synchrotrons. And the first one was the dissolved iron, so things that go through a 0.2 micron filter, indeed do travel thousands of kilometers off axis. So the transport potential is really good for that size class. This was a Nature paper, and so that was a big finding from the study. From my perspective, even more surprising, but in agreement with my hypotheses, that the particulate iron, so that meaning things bigger than 200 nanometers, also travel thousands of kilometers off axis. Do we have transport potential? Well, we have demonstrated that this is happening. This was a Nature Geoscience paper, so I guess we went down one tier in impact. But this is a really big deal for thinking about mobility of a material that folks thought wouldn't go 100 meters beyond its entry point into the ocean. So this is important. So I would say we've checked that box. Transport potential has been demonstrated. So we should be thinking about the characteristics of these materials. So let's look at what they're composed of. So in this case now, we go back to our soft X-ray spectroscopy, specifically STXM. When we look at the particles, we find iron 3 and organic carbon. So a little bit different than this first study that I told you about. But we do see still these similar kinds of large aggregate particles where carbon and iron, organic carbon, and in this case iron 3, are traveling together. They're co-located. And I think this was probably the first part of the study where we started to wish we had greater spatial resolution. You can probably see with your eye that we're not fully resolving the materials that are present, but the texture is there, it's hiding. And if we use something as simple as scanning electron microscopy, of course, these materials are aggregates of nanoscale materials. When you do the SEM in a traditional sense, in a traditional method, of course, all of that organic material that we know is present from the STXM is in a sense hidden. It's probably desiccated, and also we're not getting any chemical information from this one. So we confirmed that the particle morphology, the relationship between carbon and iron, those things were still present in this new transect, but the materials were very depleted in sulfur. And without sulfur present, iron speciation is really more straightforward. But we wanted to go ahead and have a closer look at those iron 3 particles. We can cover a lot more of our sample with a microprobe than with a STXM instrument, and we can then try to cross-reference the degree to which our observations are representative of the material. So here we're moving to a hard X-ray microprobe. And okay, this is an ugly Excel chart, but I want to, and it looks complicated in the sense of the speciation, but actually it's a pretty straightforward story. So if I can just draw your attention to these 3 bars, these are samples that were collected within the hydrothermal plume. We have good chemical information to know when we're in the plume and when we're not. And what you see here is in the blue, I've got iron 3 phyllosilicates. So these are the clays. We've got iron 3 oxyhydroxides, and we've got iron 3 oxides. So it's okay if you don't know the mineralogy or these mineral classes. The point is that it's all iron 3, greater than 90% in all of these materials within the plume. And so we decided that, given that Stixum wasn't resolving the fine scale characteristics, and microprobe was finding that the materials were quite uniform in chemistry, we decided to move up a spatial scale and do some bulk spectroscopy here at the APS in sector 20. In fact, this photograph is from the lab in sector 20. This is what grad students do in the night. They try to make glove boxes from plastic bags. And, apparently we're enjoying themselves, so that's good. But here's what the data look like. These are iron 1s or K-edge XAFs data. What you're looking at is a plot that shows samples that were collected right on the ridge axis, so that's where you're closest to the hydrothermal venting, and then we move off axis on this rough scale in kilometers. One of the notable things about these data is they don't actually change that much. There's spectroscopists in the room, you can see where the key features should be. So you'll notice that really the spectra don't change that much over the course of the transect, which was surprising to us in the sense that we expected some of these materials to alter to more stable phases over time. That didn't happen. And then the other maybe point of interest for this group is that the solid lines are bulk XAFs, and the dashed lines are from an X-ray microprobe. And so no matter kind of where we look on the sample, whether we average over square millimeters or we look at tens of square micron, we get the same answer. These materials are uniform at those 2 scales. And so what I want to do is try to, we fit those data, of course, and I want to try to summarize that for you here. So what you're going to be looking at when I show you the data is going to be organized like this. Distance from the hydrothermal vent source, moving away to the right, and then all of the data were collected from what we call the mid-plume. So that's a water column depth where we have the maximum concentration of hydrothermal indicators. So the data looks something like this. This is percent iron 3 oxyhydroxide content coming out of linear combination fitting, again moving in kilometers away from the hydrothermal vent. The station numbers are up here. And the best fit to these materials happens to be something from our database that I collected, which is iron 3 oxyhydroxide minerals precipitated in a marine bacterial biofilm. This doesn't mean that these materials were precipitated by microorganisms, but what it means is that they are materials that have been interrupted in their crystal formation. Very, very high impurity, maybe high defect, more of a 2-dimensional kind of plate type structure as opposed to a fully developed 3-dimensional oxyhydroxide. You could think of these as minerals that have been interrupted in their formation. And that signal persists out to almost 2,000 kilometers. And then as we move further out, we start to see other kinds of materials. Hornblende, that's a silicate. It's basically little rocks. It's coming from the continent. Those shouldn't form in hydrothermal vents. I think at this stage we've lost our hydrothermal signal. And then similarly, ilmenite, montmorillonite, these are materials that we don't expect to have forming in the plume. And so what we have then is this surprisingly simple, surprisingly similar across the transect material. I have to tell you that most of the places in the planet where we look, we never find materials that are this uniform. Also, materials that don't chemically transform during transport and experience alteration. So that was a surprise. But at least we know now what the hydrothermal signal looks like for this kind of plume. Keep in mind that plume is representative of a large swath of the Pacific Ocean, right? So yes, we made measurements on specific filters, specific particles, but the transect itself cuts across a really important oceanographic regime. So one of the things I think beyond speciation that we probably are almost in the process of finalizing is what is it about these materials that makes them so transportable? And what I'm showing here is just a figure from a paper that came out in 2013, written by a physical oceanographer. That he created a model for thinking about how particles settle well beyond their density. And one of the things that's super interesting that comes out of this study is that the balance of forces for composite materials can be predicted based on their fractal nature and the amount of organic carbon present. And so even though the individual materials within our particles from hydrothermal events are composed of things that are more dense than seawater, they are still quite buoyant. And it has to do with the balance of forces. And I think, our next step is probably to make friends with this physical oceanographer and see if we can add that into the modeling components so that we can get better estimates of how much iron might be mobilized and transported by this mechanism. So I would say that's a work in progress. So I'll just start to wrap things up here. The hydrothermal plume particles, no matter where we look, are pretty consistently composite materials. And they are composites of organic plus inorganic materials. They have both crystalline and non-crystalline precipitates. And the love story, the main story, is that carbon and iron travel together in these systems, and it is probably the organic carbon that makes the transport possible. So yeah, we've got leaky vents, and we've been able to show it in a variety of ways. And it's this composite nature with organic matter that's keeping particles from settling and allowing that transport to happen. So let's go ahead and do a little wrap-up. Don't want you to forget that Earth materials are complex and heterogeneous. This is the main reason I think synchrotron tools are so important for advancing our understanding of these materials and how they play into the biosphere and the hydrosphere and the geosphere. We use the synchrotron tools, of course, to further our hypothesis-driven science funded by NSF. But in reality, in practice, we generate discoveries by using these tools to look at materials. And if I go back to my multi-modal argument, I just want to leave you with this idea that Earth and environmental scientists over the past 10, 15 years, we're already doing this. We're already investigating multiple elements, spatial scales, and properties. And this is a suite of capabilities that we hope that all of the US synchrotron facilities will continue to invest in and cultivate. And we're not the only user community that benefits from this general approach to synchrotron science. And then, we're still struggling with some things. We would like more high throughput, but then honestly, we're not super well equipped to deal with those data streams. And so there's some growth that needs to happen in my community, and hopefully that'll be a partnership with the synchrotron facilities. What would up our game? I always get in trouble when I say that a lot of my experiments are detector limited. We can usually get more photons than we know what to do with, more photons maybe than our sample can handle. More photons are good though because it furthers instrument development, smaller spot sizes. I'm not complaining, just saying that a lot of times we find that we don't have quite the right detectors to take advantage of all the photons, and especially when we're dealing with ocean systems where our concentrations are low, this is something we struggle with. As we deliver more photons to the sample damage becomes increasingly a bigger problem for our community. I had great conversations in Sector 13 today about, can you measure your data faster? Can you keep your samples cold? Right? There are strategies that people already use to try to address this. It will be an ongoing challenge. One of my mentors, Alain Monceau, likes to say the future is fast and cold. That's part of the scenario here. And going from those general properties and challenges to, wow, I would love more tender X-ray STXM instruments, I'll just put that out there. They would up our game scientifically. And then that colloidal size fraction is a super mobile, super chemically reactive class of materials. And I would say as we move toward imaging and spectroscopy down to the bottom of that size fraction that we actually still consider something a particle, this is going to open up big new areas of discovery and research for our field. And there's really a seventh factor, I think, in the work that we do as earth and environmental scientists. We know that the science leads our choices. It leads what questions I ask, what grants I apply for, what beamlines I want to use. Science is really the driver, and I know that's true also for facility upgrades, instrument upgrades. We always need to know what science is coming down the pipe. But I think we always need to remember that people do the science. And so the collaborations with the beamline scientists, the synergy between the science and the instrument development, these are key. And I want to think more, and I'm thinking a lot more, about which instruments are going to be available for my students and my students' students, and what kinds of unanticipated discoveries are they going to be making in the future. So anything we can do to support the development of that future user pool benefits all of us. And many people help us do this work, and I'd be happy to take any questions that you might have.
You have a microphone, so okay, speak up.
So thanks, Brandy, for the talk. I wanted to ask two things about iron transport. One is, in some of the pictures that you had showing the concentration with location, there seemed like there was almost a gap. Is that just because it was sideways current that didn't get your sampling on?
Yeah, good eye. That gap in the plume is definitely, well, the physical oceanographers are telling us it is because there is a cross-cutting circular current at that location, so we have a gap. Sorry, I think I'm making some feedback. I'll stand back. So yes.
Okay. Yeah, and then the other thing is, I wonder about transport of iron into the ocean from dust. You mentioned that. Is that a way off scale below these other contributions?
No, and in fact, I would say it's important. In different locations around the global surface ocean, it's definitely going to be important. And I would say it's been better studied, and it is not a controversial source. In terms of what is the actual balance from a global budget perspective, I think those numbers aren't really known. I think the error bars on the dust input category, even though it's an established and accepted source, are really huge. So I think that's something coming in the future. And we don't have good error bars either on the estimate of transportable hydrothermal iron. I think folks didn't want to spend a lot of time on it until we could demonstrate that this was something that they should be thinking about. I'm trying to stand somewhere where I feel like I'm getting less feedback.
Hi.
Hi.
Quick question. Do any of the hydrothermal transects, like the one you did, intersect with high nutrient, low chlorophyll zones? And if they do, how do they compare?
Yeah, that's a great question. So you might have noticed that transect began off the coast of Peru. And so there's a big upwelling zone there where you have also an oxygen minimum zone that forms because of the upwelling of nutrients. And so the transect cut across that, then through some open ocean, and then over the hydrothermal vent. And there are, it depends on what water depth you look at, but one of the interesting things about that oxygen minimum zone, and we did work on it in collaboration with Phoebe Lamb using similar techniques. One of the things that's pretty fun is that the particulate iron is in the chemically reduced form. and oxidized, despite the lack of oxygen. So there's internal redox cycling, maybe linked to the nitrogen cycle, but at one point that oxygen minimum zone is so intense that actually even the sediments have iron too. It just goes right down to the bottom of the water column. So there's different iron chemistry. Like when you get down to like what mineral phase is there in terms of solids, they are different groups of oxyhydroxides.
Thank you.
Why, you said that you use helium-3 as a marker. Why helium-3?
So I don't personally make those measurements, but the way that the chemical oceanographers like to use it, it's a mantle helium source. And so it has a distinct isotopic signature that can be linked back to that source. Does that make sense? And it's a dissolved gas. It doesn't tend to be very chemically reactive, and so it's a really good conservative tracer.
Yes, go ahead.
The bacteria that you talked about being active in the vents, aerobic, anaerobic, sulfurine?
All of the above. Yeah, so the microbiology at the hydrothermal vents is not really something that I got into, but if there is a redox reaction to be coupled, it's happening. Every time folks look hard enough, they can find it.
Any other questions?
So between the different vents that you looked at, is the size and characteristic of the particles similar, or are there some vents with different sizes and different temperatures? I know nothing. So.
Yeah, there is variety. I would say, let me start with the sort of unifying characteristics. The degree to which materials aggregate, that's a really common characteristic of the materials. And we've probably looked at materials from, I want to say 8 different vent sites. And oftentimes we're doing these studies where we use robots or submersibles, and we know exactly where those materials are coming from. The fluids, the temperatures, the chimney, the host rock, the geophysics, the whole business is known very well. And so we can think about a wide variety of different kinds of fluid compositions leading to some similar characteristics across these materials. What differs, I think, is which biota live in the area, how productive they are, how much floating particulate organic carbon is available to mix with those vent fluids as they're coming up, which microorganisms are present, and which metabolisms are they carrying out. I think those differences then tell us, the degree to which we expect our sulfur to be, our organic carbon to be sulfurized. That affects the metal chemistry. But I think those are details in the sense of, like, if we care about iron getting away from vents, that transport potential is there at all the sites that we've looked at so far.
I have a question. From the time that you collect data to the time that you go to a synchrotron, are you worried about things changing in the samples?
That is a really good question. So we spend a lot of time shipboard protecting our samples from ambient temperature and oxygen. And so what that usually looks like for us is if it is a cabled filtration system, as soon as that comes up onto the ship's deck, that little filter housing goes directly into an anaerobic glove bag. Those samples are processed however they need to be. Note that the glove bag is also in a HEPA bubble, so we're trying to minimize contamination of particles as well. And then those samples get packed under nitrogen in mylar, often frozen, but not always. And then we just have to kind of babysit them until we get back home and do our synchrotron time. So we think about temperature and exposure to oxygen as being probably the 2 most important artifacts, but we would like to analyze them as quickly as possible.
So ideally you would want to have a synchrotron on a ship, right?
Yeah, we want to have a synchrotron at the seafloor. That would be the best.
Any other questions or comments? If not, let's thank Professor Toner once again.